Hey, I'm back for the bacteria, and this one's gonna be a little bit shorter, but there are two main points that need to be understood about bacteria to finish the chapter on bacteria and archaea, and it's how we classify them basically on the tree, and also how they do gene or DNA gene transfer, right? So let's talk about the classification first. We rely on metabolism. So for bacteria, depending on what you're doing, so the cyanobacteria, nitrogen fixing, bacteria on skin, digestive tract in yogurt, or decomposing banana peel, you are following different metabolic pathways. If you're cyanobacteria, you're autotroph and you're aquatic. If you're a bacteria decomposing banana peel, you're going to be heterotroph, right? Then there is an additional layer of that is aerobic versus anaerobic. So do you use oxygen at the end of the electron transport chain of cellular respiration or not? Something else. And that's conditioned by the environment. You can imagine that in our guts there is not a lot of oxygen, so it's usually obligate anaerobe, so not using oxygen as an electron transport acceptor at the end of the electron transport chain, or facultative at least anaerobe. So they can just switch. And you have also similar things that happen with bacteria in yogurt that are doing fermentation. Now, in the environment, it's also possible to find anaerobic bacteria. And so if you look at this particular diagram here, we talked about the digestive tract versus the surface of a mouse. It's the same for us. But we need also to account for when the waterlogged soil is present. If water is saturating, all of a sudden you don't have a lot of gas exchange that's possible, right? Same thing at the bottom of a lake or a river. So you can have river, it's a little bit less because you have a current, right? But at the lake you can have sedimentation, and in those or deep water there's not a lot of oxygen. So that's important to keep in mind. Keep in mind that we are classifying bacteria along the metabolic pathway they're using and what they're doing, right? So decomposers, heterotrophs, autotrophs, all of those things. But we also have to keep in mind the environment can be changing. So those population can boom and then bust if you're an anaerobic bacteria. And conversely, the aerobic bacteria can boom in terms of population and then bust when the water saturates the soil, right? So with that being said, how do bacteria reproduce? We already know it. It's called binary fission. So we have seen it in the past, right? It's not the first time. It's not a first rodeo on binary fission here. So it's a simple DNA replication followed by a cytokinesis, right? Event. Binary fission, pretty straightforward. Now there are other ways to transfer DNA, and they do it horizontally. So the horizontal DNA transfer can be of three different types: transformation, transduction, or conjugation. So let's focus on the first one, transformation. In transformation, some DNA is in the environment. It can be due to the cell dying, right? It can be due to a release of a viral particle that leads to the death of a bacteria. It can be due to simple, striking light strikes or light strike. A storm, someone stepping on them, like anything that leads to the death of a bacterium can lead to DNA being released in the environment, and the DNA can be uptaken readily by another bacterium. And that's not just viral bacterial DNA. It can be any type of DNA that's out there, so they can incorporate it. That would be called transformation. When we talk about transduction, here all of a sudden we're talking about a virus infecting a source cell, and in the process of that infection, incorporating part of that DNA of a source cell into the newly formed assembled viral particles, right? So that arrow here considers attachment, penetration, synthesis, assembly, and release, right? In the synthesis part of that arrow, some of the host DNA got incorporated, right, into the viral DNA. So now when the virus goes to the next bacterium and infects it, it injects itself in there, the DNA, and that DNA will contain the source DNA from the host, from the source cell. So that assumes that first infection is lytic pathway, right? Because the viral particles are released and kill the bacterium. And the second one is potentially lysogenic, because you understand that otherwise that cell is just going to make more copies and die. So the way we're talking about transduction is from the point of view of this bacterium uptaking DNA from another source cell, another bacterium, right? And that's via modulated by the viral particle. So we're not really talking about the viral infection here. We're more talking about the source DNA from the host, the first host, to the next host, right? It's really important to see the subtleties, because that recipient cell is not about the viral DNA. Transduction is about the bacterium DNA from the source cell coming to another cell, another bacterium, thanks to a virus. We're getting a ride on the virus, if you want. So that must be lytic pathway here and then lysogenic pathway there. That makes sense? And the final one is conjugation. Is that when the sex pili come back. So sex pili, or in that case pilus, because there's only one, two bacteria get next to each other, and they connect with the sex pilus. And when that connection is done, a plasmid from one can be transferred to the other one after replication of said plasmid. So it's bacteria sex, if you want. That's when they get next to each other, transfer some DNA. It doesn't have to be a complete transfer. It can be interrupted, but that's another type of horizontal gene transfer. So you have two types of gene transfer: a vertical one, binary fission, and you have 3 horizontal ones. Important that you layer in that arrow here all of the steps of a viral infection, and keep in mind that this transduction is about the DNA of a bacterium to another bacterium. Transformation is uptaking DNA from the environment that can follow the death of the bacterium for any reason, or other pieces of DNA out there. Okay? And remember, that is not how we don't classify bacteria based on how they do transformation, transduction, conjugation. They all do all of those. We classify them based on metabolism, and it's linked to the cellular respiration pathway for usage of oxygen, and it's also linked to metabolism such as autotroph or heterotroph. So do you do chlorophyll based or non-chlorophyll based photosynthesis, or do you not, right? And with that, we'll stop it there, and we'll see you in the next one. Take care.